Good morning or good afternoon, everyone, depending on where you are joining us from, and welcome to today's webinar. My name is Eliza Berry from Business Review, and I will be your host for today. It is our pleasure to have Biogelx Limited with us today, who will be presenting today's webinar titled Modeling in Vivo Conditions in Vitro, 3D Hydrogel Systems for Biomedical Applications. Today's guest speakers are Robert Edward Schwartz, MD, PhD, Assistant Professor of Medicine at the Stanford Medical College of Cornell University, Professor Matthew Dolby, Professor of Engineer at the University of Glasgow, and Mitch Scanlon, CEO of Biogelx Limited. I'd like to welcome you to our platform on24. You'll notice that this webinar is browser-based, so if you disconnect for any reason, please just click on the link that you received via email to rejoin the session. In order to ask questions, you can send them in via the questions widget. Just type them into the box at the top left-hand corner of your screen and click Submit. We will allocate some time at the end of the session to address any questions or thoughts that you may have. Please use the yellow Help widget if you require any assistance, and you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. But now, please allow me to welcome Mitch.
Thank you, Eliza. My name is Mitch Scanlon, and I'm just going to spend a few minutes just introducing the webinar and just providing a brief overview of the Biogelx synthetic peptide hydrogel technology that we've been developing over the last 5 years. So the key learning points that we'd like to try and get across in this webinar are, to give you an overview of the hydrogel technology. But also, Matt and Rob have both been working with this technology in their research groups, and I'd like them to present some of the case studies and applications that they've been developing in the areas of using the hydrogels for differentiation to improve cartilage phenotype in 3D culture, and also in vitro liver models. So Biogelx, spun out of Strathclyde University in 2013. As I said, we're developing a proprietary synthetic 3D peptide hydrogel technology. And I think there's increasing evidence, and it's becoming widely acknowledged that 3D cell culture techniques are more physiologically relevant than traditional 2D models. And as a result, biotech and pharma companies are rapidly adopting these systems in a range of biomedical research applications. Ultimately, they're trying to create improved and more predictive data that can be modeled in vitro. Hydrogels, including synthetic hydrogels, are emerging, I think, as one of the most promising alternatives in 3D cell culture applications, because they mimic the salient elements of native ECMs, extracellular matrices, and they also possess mechanics similar to those of many soft tissues. Having said that, I think the industry recognizes that there is a need to identify and develop new improved 3D models, and some of the key requirements that the industry are looking for are models that have enhanced cell-to-cell interactions, and this is really dictated by the stiffness of the extracellular matrix. They're also looking for models that have good interaction between the cells and the extracellular matrix. Biocompatibility, the ability to be able to grow cells, is obviously critical, and this, with respect to things like reproducibility, ease of use, scalability, printability, I think this is an area where, in particular, the synthetic materials that are being developed, have some advantages. From a biocompatibility perspective, certainly some of the natural hydrogels, that dominate the market at the moment, are very, very good at growing cells, but when you want to start to have reproducible materials that are going to be consistent and easy to use, and as these applications mature and scale up, I think synthetic materials potentially have a key role to play. So just very briefly, the Biogelx technology platform is basically based around simple peptide technology. It consists of a gelated peptide, a hydrophobic gelated peptide, and a hydrophilic surfactant peptide, which in the presence of water will self-assemble into nanofibers that consist of a hydrophobic core and a hydrophilic surface coating. These materials, in this format, we refer to them as a pre-gel, and the gelation process is caused by the addition of divalent cations, which are typically found in most cell culture medias. And you'll see on this slide there's a few images. The small image in the middle shows you what this extracellular matrix, this synthetic extracellular matrix looks like, and is very, very similar to what you would see in a natural system. To the left-hand side, this is from the work that Matt's been doing at Glasgow University. This shows a mesenchymal stem cell that's growing on the matrix, and as I mentioned before, it's very important that the cells and the matrix are integrated and work well together, and you can see that level of integration in the image here. And on the right-hand side, one of the advantages of this material and one of the key differentiators is the ability to be able to mimic a whole range of different tissue types or extracellular matrix, and this is done by adjusting the peptide concentrations to create different stiffnesses of material for research purposes. So as I said, this is the ability to be able to adjust the gel stiffness is one of the key attributes of this material. And this is very simply done by, the material is provided commercially as a lyophilized powder. So simply by adjusting the weight of powder that you use and mixing different concentrations with water, we're able to create these different stiffnesses of tissue that can mimic very soft tissues such as brain or liver, through to stiffer tissue types such as cartilage and bone. As I mentioned earlier, one of the challenges with synthetic peptides has been the interaction between the extracellular matrix and cells, and this has been a sort of key focus of development here at Biogelx for the last few years. And what we've done in order to address this and ultimately to improve cell growth within these synthetic materials has been to identify the materials that naturally cause cell attachment and encourage good cell growth, such as laminin, fibronectin and collagen, and we've been able to identify and select synthetic sequences for these materials and to integrate these into the synthetic technology platform. So what we now have is a totally synthetic platform of functionalized gels that are functionalized individually, and we're also looking at some combination functionalized gels, which are now showing some really, really good cell growth data, comparable with the biocompatibility data that you would see with some of the natural synthetic materials like matrigel. So, in summary, I talked about some of the sort of key requirements for 3D materials moving forward, and I think the synthetic material that we've developed at Biogelx ticks a lot of the boxes, and certainly in terms of the range of stiffness, reproducibility, and printability, the ability to be able to use these materials increasingly as the 3D bioprinting market expands, at a significant rate. The ability to be able to take these materials and to print them into high throughput automated or precision applications will become critical. We've got one polling question during the webinar today, which is really we're just curious to find out from the audience what you consider to be the most important criteria that's driving your selection of 3D models. And I would just ask the audience, you can select more than one of these if more than one are relevant to you. But if you could just take 30 seconds just to review the list and just flag which are the key elements that are driving your selection criteria for choosing 3D models, we would appreciate that. The next slide will show a quick summary of that analysis. So I'm waiting for our organisers to just update the results of that poll. Okay, so the results are coming through now. So it's really interesting. It's interesting as a supplier of these materials. Reproducibility is the one that's coming out. Has polled 40% of the votes, and I think that's probably relates to the fact that a lot of people today are using naturally derived hydrogel materials, and these can be notoriously unreliable from one batch to the next. And it's interesting that's been highlighted. Interesting that nobody has ticked biocompatibility, which I suppose as a developer of a synthetic material, biocompatibility has been a key focus for us and has been a real driver for certainly the functionalization. So I'm going to these results will get published and be part of the webinar. Is posted afterwards. But I'm going to now hand over to Matt Dolby, and Matt can tell you a little bit about the research that he's been conducting in his labs in Glasgow.
Great, thanks, Mitch. I'll just need to work out how to scroll on this slide. There we are. Following on from Mitch's discussion about the importance of reproducibility and also for synthetic gels, I'd like to tell you a little bit about the work we've been doing in our laboratories here in Glasgow in conjunction with Biogelx. Particularly, I want to talk about two things. The use of peptide hydrogels to direct stem cell differentiation. This was not developed in Glasgow, but we developed a little turn on this that I think is important for synthetic gels, and that is trying to direct stem cell differentiation and also look a little bit about possible discovery of drug types. And then the use of peptide hydrogels to improve the cartilage phenotype in 3D culture, which again is a big regenerative medicine challenge. So for the first story, I'm going to focus on the use of pericytes. So let me first explain what these are. So these are perivascular stem cells, which are considered multipotent precursors of mesenchymal stem cells, which of course most people have heard of now. Pericytes are found on the vasculature throughout the body. They're multipotent as a mesenchymal stem cell, so can turn into osteoblasts, chondrocytes, adipocytes, fibroblasts, etc. And they were first published as multipotent precursors of MSCs in this paper in Cell Stem Cell from the Pio group over in Edinburgh in 2008. And we wanted to observe differentiation on different gel stiffnesses. It started as quite a casual study. We wanted to look at the very well-known Adam Engler, Dennis Disher study published in Cell in 2006, where they put mesenchymal stem cells onto gels of 3 stiffnesses. They had a very soft gel around about 1 kilopascal, a medium stiffness gel of around about 15 kilopascals, 10 to 15 kilopascals, then a stiff gel, a rigid gel, up to around about 40 kilopascals. And on the soft gel, around 1 kilopascal, they saw expression of neural markers from mesenchymal stem cells. On the medium gel, the 10 to 15 kilopascal gel, they saw myogenic expression, muscle cell expression for the mesenchymal stem cells. And on the very rigid gel, the 40 kilopascal gel, they saw expression of the osteoblast phenotype. Now, just a little aside, the observation of neural expression markers is kind of a moot point. And personally, I believe this is the expression of stem cell self-renewal markers. So the bone marrow, as for neural tissues, is very soft. It's just less than about a kilopascal in stiffness. And mesenchymal stem cells or perivascular cells within the bone marrow will express neural markers like beta-3 tubulin and nestin. And these are usually considered to be when they're homing hematopoietic stem cells. So they share the bone marrow niche with the hematopoietic stem cells, and the mesenchymal stem cells and pericytes are thought to have a supportive role. And actually, Bruno Pio and his group have shown that if you culture hematopoietic stem cells over pericytes rather than mesenchymal stem cells, you get better self-renewal of the pericytes. But this is a whole different story for a whole different day. But the important thing is we could take 3 stiffnesses of gels, the soft, the stiff, and the rigid, and we could differentiate the cells. And for the soft and the rigid, we saw the same as Engler, with the pericytes turning into cells expressing neural markers on the soft gel, and here you can see expression of beta-3 tubulin in part C of the image on the right. And on the far right in part E, on the rigid gel, you can see expression of bone markers, in this case RUNX2, here indicating differentiation towards osteoblasts. We differed in the middle, whereby when we used our stiff gel at 13 kilopascals, we didn't see myogenesis. We saw expression of chondrocyte markers, and we've given an example here in Part D of SOX9 having increased expression. So for the most part, we agreed, but apart from this one in the middle. Okay. So what we wanted to do, the big advantage we had over other groups at this time was that we're using a gel source that were very similar. So we had 3 highly controlled rigidities, and that's really important. Using many material systems, controlling rigidity or stiffness with good level of control is quite tricky. With the Biogelx gels, it's pretty straightforward. But because the gels are synthetic, and the only thing that's changing is the fiber density of the peptide fibers, actually they're chemically very similar. We don't have to coat the gels to get the cells to stick. They'll stick onto the native gel, so there's no intermediate layer to add complication. And we don't have to use cross-linking agents such as glutaraldehyde to get the most stiff gels. And this gives a big advantage because at the same time my group was getting into looking at metabolites and metabolomics using mass spectrometry. And we wanted to find metabolomically quiet material systems to tell the cells what to do, but not to affect the metabolites themselves. So we just wanted stiffness to tell the cells what to do, not the chemistry or the cross-linking agents that we're putting into the gels. So as the cells differentiated, and here I'm just going to present on chondrogenesis and osteogenesis, because the neural story, whilst it worked, is more complicated, and so it's better left when there's longer time. What we did, we took 2 time points, an early time point and a later time point during the differentiation process for chondrogenesis and osteogenesis, so on the stiff gel and on the rigid gel, and we followed the metabolome with time. We looked particularly at the lipid metabolome, because this is where we got the most hits. And it's an energy-dependent, these are energy-dependent metabolites, so and differentiations, an energetic process, and so energy seems a good place to look. And what we look for was unique depletions with time. So metabolites the cells seem to be using to differentiate. And so at the top of the figure in Part A, we can see that this metabolite, GP18-0 or lysophosphatidic acid, is being used by the cells when they undergo chondrogenic differentiation, so on the middle stiffness hydrogel. Whereas it's not found whereas cholesterol sulfate, which I'll tell you in a second, is not found in this gel. So this GP18-0 is a unique depletion. When we look at the rigid gel, so the parasites undergoing osteogenesis, we see the opposite. There's no change in GP18-0, so this isn't part of the osteogenic process. But for cholesterol sulfate, we see this depletion between week 1 and 4, which suggests this is being used during the osteogenic differentiation process. And if we use pathway building software, we can see that GP18-0 is a glycerolipid, whereas cholesterol sulfate is involved in steroid biosynthesis. So all we've done is we've used the gels to let the cells differentiate, and we've just looked at what the cells are using to differentiate in terms of metabolites. And metabolites, as I'm sure you all know, are the small building blocks of biochemistry, so little bits of biology that cells use to drive their biochemistry as they differentiate or as they grow. So the next thing we did was culture the cells just on glass cover slips. What we wanted to see was, could we take these metabolites the gels had showed us, and could these metabolites then themselves drive cell differentiation, just individual metabolites? And here we use both parasites, as you can see in panels A and E, and mesenchymal stem cells, as you can see in C and G. So on the left hand of all these images, you can see the control cells. So these are just parasites or mesenchymal stem cells cultured on a cover slip. In green are phenotypical proteins. So on the left, we can see osteopontin and osteocalcin. These are proteins expressed during bone formation. On the right, we can see aggrecan and collagen 2. These are proteins expressed during chondrogenesis or cartilage formation. So if we take the figures on the left to start with, we can see the control cells where there's no green being expressed, so no bone marker proteins being expressed. Then in the middle, we use osteoinductive medium, OIM, which is dexamethasone, glycerophosphate and ascorbic acid to drive bone formation. You can see good expression of osteopontin and osteocalcin in both the parasites and the mesenchymal stem cells. But then in the last column, we've just used cholesterol sulfate by itself. So this metabolite that we identified just by following the fate of the cells on the hydrogels. And we can see that we're getting similarly efficient expression of the bone phenotype, the osteogenic phenotype from stem cells, as if we use the more complex and standard osteoinductive media. Remember that dexamethasone is a powerful corticosteroid. Over on the right, in the right-hand panel of images, this is the chondrogenic panel. We can see again on control, on the left image of this right-hand panel, we see no expression of aggrecan or collagen 2, these chondrocyte markers, whereas in the middle, where we use chondroinductive media, and this contains transforming growth factor beta, TGF-beta, insulin, and dexamethasone, so corticosteroids and growth factors to drive the chondrogenic phenotype. Whereas over on the right, where we've just used this single metabolite, GP18-0, we can see similar levels of chondrogenesis in the stem cells, both the pericytes and the mesenchymal stem cells, as when we use the very complex, expensive chondroinductive media. And we find this kind of interesting because things like dexamethasone and growth factors, they can have off-target effects when they're used in the body in vivo. And so single metabolites that are found in vivo anyway might seem a simpler, more cost-effective and potentially safer way of inducing different phenotypes in vivo. So the take-home message from this first study is that these gels we've used are pretty much identical. We don't have to differentially cross-link. We don't have to use chemicals to induce differentiation or coat with proteins. This has to be done with, for example, acrylamide systems. This means that they are metabolomically quiet, vital to allowing us to look for the metabolite differences caused by the gels alone. This study gives us a demonstration that gels can be used, that biomaterial systems can be used to persuade stem cells to do things, so you can watch them and identify bioactive metabolites that could potentially go on to be developed as drugs to control stem cells. And this work was published in Alakpa et al. In Chem in 2016, tunable supramolecular hydrogels for selection of lineage guiding metabolites in stem cell cultures. So moving on to the second story, chondrocyte tissue engineering. Now, with an aging population in developed countries, we're particularly worried about musculoskeletal disease, arthritis in particular, because it costs, it has a large socioeconomic burden, people are in pain, we're all living longer, and as we have this increased lifespan, we also want to enjoy increased health span to go along with this. So we have to think about regenerative medicine approaches to helping solve problems such as arthritis. And because the stiff hydrogel, the medium stiffness, 13 to 15 kilopascal hydrogel, could induce chondrogenesis without using differentiation factors, we decided to have a look at this. This time, I should say in the previous study, we were putting cells onto the hydrogels rather than in the hydrogels. We're just using the substrate, but here we're putting cells within the hydrogels as a three-dimensional tissue engineering construct. I should mention we didn't use a solid 24-well plate-fitting hydrogel as shown in part B. We used droplets of hydrogels, so little spheres of hydrogels with the pericytes inside. So we're still sticking with the pericytes here. We should note that there's some parallels between the stiffness of the hydrogel and the stiffness of chondrons that chondrocytes grow on in the cartilage environment anyway. So when we encapsulated the pericytes, we saw, as we can see here in part A, very good cell viability. All the cells were alive over all the time course we did, up to 168 hours. And further, by qPCR, as we can see in part B, we saw enhanced expression of chondrocyte markers SOX9 and collagen 2 after 1 week of culture. And if we look at histology after 28 days, we can see in part C and D high levels of expression of aggrecan, as shown in C, and collagen 2, as shown in part D, both stained green with the nuclei stained blue within these hydrogels. So we can see we're getting both viability, survival of the pericytes, and differentiation of the pericytes in 3D, as we'd seen on 2D with the stiff hydrogel. So we wanted to move on to looking at different markers, and so we moved to using qPCR at various time points from day 1 up to day 35. Here we used planar control, which is just a cover slip. We used alginate as a typical material that chondrocytes are encapsulated in, a gold standard if you like. We've used the stiff hydrogel, which is denoted here in the grey bars as Fmoc F2S. And we've also used the hydrogels, but with contra-inductive media. So that's denoted with a positive sign. So just the cells in the hydrogel alone with the negative sign, cells in the hydrogel with chondrogenic media with the positive sign. And what we see is in the hydrogels, compared to alginate, compared to cover slips, we get much better expression of SOX9, of collagen 2, of aggrecan, and of type 10 collagen, as would be expected for cells undergoing chondrogenesis. And really importantly, much higher expression of cells in alginate in 3D. As you can see by comparing the grey and the yellow, the hydrogel bars to the orange alginate bar. But whilst it was noted that adding in the contra-inductive media increased chondrogenic differentiation further, it's important to note that the hydrogel itself, the stiff hydrogel itself, drove chondrogenesis just fine. Actually, it drove it rather better than the using contra-inductive media with the hydrogel. If we look at the ratio of proteins, so we looked at GAG to collagen 2 and also collagen 2 to collagen 10. Now, the important one really is to look at the collagen 2 to collagen 10, because if we consider the structural function, the structure of cartilage, at the top you've got the superficial zone. This is the articulating hyaline surface of the cartilage that glides and allows the joints to move, and this is what gets worn out during arthritis. At the deep zone of cartilage, close towards the bone, you've got hypertrophic chondrocytes and mineralized cartilage as it interfaces with the bone. Now at the top, at the articulating surfaces, the cells should be high in collagen 2. At the bottom, at the collagen 10 surface, at the bottom surface, the bone interfaces should be high in collagen 10. So if you look at the graph in part B here, you can see that the cells grown within the stiff hydrogel alone, without the contra-inductive media, have a much higher collagen 2 to collagen 10 ratio, which means we're getting this more hyaline articulating cartilage phenotype within the gel compared to using TGF-beta in the inductive media. And it's very well known that TGF-beta causes overexpression of collagen 10 in cultured cells. So this is quite an important observation. So if we look at the metabolome, we can see very clearly on this figure below, if we see the planar cells grown on planar samples compared to cells grown within the hydrogels, very large difference in the kind of heat map from looking at the metabolome, the cells from planar versus undergoing chondrogenesis. The cells within the gels, but with and without the contra-inductive media, look pretty similar, except for discrete pockets of differences, as you can see outlined by the dotted lines. And it's within these pockets that we're researching now, because this must be the areas where we have the difference between collagen 2 expression and collagen 10 overexpression. So we have to research these more deeply as we go forwards. But for the last couple of slides, just to summarize, for techniques such as matrix-assisted autologous chondrocyte implantation, or MACEs, I'll quickly show in the next slide, use of TGF-beta in differentiation causes chondrogenic cells with high collagen 10 expression, typical of deep zone chondrocytes. That's not what we really want when we are repairing cartilage. The gel alone is allowing us to generate chondrocytes from stem cell populations with higher expression of type 2 cartilage, typical of hyaline articulating cartilage, and this work was published in the LACPA in Scientific Reports in 2017, improving cartilage phenotype from differentiated pericytes in tunable peptide hydrogels. So just for the last slide, it's just a picture of the structure of cartilage. On the left, we can see the part 1, the superficial zone. This is the articulating zone. Then at the bottom, you've got the larger hypertrophic chondrocytes, high in collagen type 10 in the calcified zone, whereas in the superficial zone, you have a higher amount of collagen type 2. In the middle, from the polymers reference, this is a figure showing MACE, matrix-assisted autologous chondrocyte implantation. So on the knee, you can see a cartilage lesion. They take a biopsy from the non-articulating part of the cartilage, expand these up in culture as chondrocytes, then they can inject them back into the wound site. That's an autologous chondrocyte implantation. Or they can use a polymer to fill the wound site first and then inject the cells in. And this is a matrix-assisted autologous chondrocyte implantation. They put a little patch over this. This is a shaved patch of periosteum. There's an oval patch depicted on the bone just below. They shave that off. It's like a skin, and they suture that over the defect. And on the far right, you can see an image of Carti patch being put into place within a knee. And this is an alginate agarose scaffold. And we can see from our data that alginate doesn't push the cells to become chondrocytes, or doesn't support them to chondrocytes. So if we think about towards the future for regenerative medicine using defined hydrogels with a stiffness that tells the cells what to do, we can probably get a much better repair going forwards. So that's all I want to tell you today. Thank you very much. So I'm handing over to Rob now.
Well, thank you for that. Want to get started here and really just want to provide, basically just give some motivation for what has been driving our thought process and our research, with ultimately the goal to generate basically mini livers that could either be used for modeling human liver disease, phenotype and function in vitro, with the ultimate goal to be able to transition these for use in vivo. And basically, one of the major challenges that has existed when people have wanted to work with hepatocytes or liver cells in vitro is that they rapidly de-differentiate, meaning that they rapidly lose their phenotype and function. And as a consequence, because it's been so difficult to maintain both the survival of liver cells as well as the phenotype and function of these cells in vitro, it really has created significant challenges to use and study hepatocytes in vitro. And, when we really think about this problem, then, this is really surprising, because if you guys know about the liver itself is a very unusual organ, such that when the hepatocyte, the parenchymal cell of the liver, is injured, basically either through toxins or drugs, the hepatocytes that are lost, the liver cells that are lost that die, are replaced rapidly through the proliferation of undamaged hepatocytes. However, when this process occurs too acutely in vivo or occurs chronically, it can lead to chronic liver diseases. And so, this is a huge problem in the United States, Europe, and throughout the world. Basically, over 20 million Americans and 29 million Europeans live with some form of liver disease. 3 million Americans and about 6 million Europeans live with the end-stage process of these liver injury processes. And as a consequence of that, 14,000 Americans are currently listed for liver transplantation. About 8,000 Americans and about 6,100 Europeans are transplanted each year. And as a consequence of this, about 31,000 Americans die each year from acute and chronic liver disease. It's the 12th leading cause of death in the United States. In Europe, about 85,000 Europeans die each year from acute and chronic cause of liver disease. It's the 5th leading cause of death overall in Europe. And worldwide, over 1 million people die yearly from cirrhosis. So if we were actually able to build a structure, a tissue that could actually maintain the differentiated function of liver cells, of hepatocytes, that prevented their rapid de-differentiation, maintain their function, maintain their survival, maintain their phenotype, you can imagine that these could be used in a variety of different applications, such that for both clinical practice and basic science. And more specifically, in the clinical arena, you can imagine that we could use these for drug testing, for the use in a bioartificial liver in vitro, or directly actually for transplantation as a construct in vivo. Now, in vitro, if we're able to maintain these cells long-term, maintain their function or phenotype, we can then use them as models to study both metabolic liver disease, hepatotropic disease processes, and also potentially as models to study both hepatocyte function and development. Now, when we think about the different components and the limitations that exist in hepatic tissue engineering, I've already kind of alluded to some of the major challenges in working with the cellular component of the liver, the hepatocytes, that they rapidly de-differentiate. And the question is, what is the major driver for this process? And in a large body of literature that I'm not going to present today, basically what we and many others have shown is that the microenvironment, the environment in which these cells are sitting, actually dictate through both physical signals as well as chemical signals to the cells, and it's through these signals that help maintain the differentiated phenotype and function of hepatocytes. And so, if one were to drill down or go down more deeply, it's really these microenvironmental components that are really important in actually driving and maintaining hepatocyte function. And so it's actually, if you take these all together, the phenotype and function of the cells are actually going to be dictated by how we construct this environment. So basically, it's, as I've mentioned already, it's really important to try to include components of the hepatic or the liver microenvironment that would then mimic what we see in vivo, but now in vitro in a hydrogel. And many groups have tried a variety of different natural, non-synthetic approaches. And what was mentioned by Mitch, there's a lot of both variability and reproducibility issues with doing this. The other problem is the natural hydrogels, such as in Matrigel or collagen, don't readily recapitulate the elastic modulus or the stiffness that is what we find in vivo. And so basically, one of the advantages that attracted us to work using these peptide hydrogels was, one, that we can easily functionalize these gels with peptide motifs that are found in vivo in the liver, specifically RGD, and that this peptide can be easily incorporated into these hydrogels. Moreover, the stiffness of these hydrogels are actually quite similar to what is found in normal liver, as opposed to the variety of natural hydrogels, which actually the elastic modulus or the stiffness is significantly in orders of magnitude higher than what we find in vivo, and as a consequence contributes to some disrupted expression of a variety of different markers, as well as significantly impacts the function of the hepatocytes. So what I want to then show you here is we can then take gels that are made with or without this functionalized component, the RGD peptide motif. We can then rapidly make these gels with the hepatocytes embedded within them, and basically then show that these gels can maintain both their structure over time in vitro. And we're showing this here for 5 days, but we've actually been able to show that we can maintain their structure for several weeks. And this is just drilling down now, just microscopically here, using atomic force microscopy, showing that these peptide components, these motifs, are actually incorporated onto the surface of the hydrogel. And that when we then incorporate both hepatoma cells, so a liver-like cell line, HepG2, or primary human hepatocytes, we can actually produce both little spheroids from these. When we embed those spheroids within the hydrogel, incorporating this peptide motif, we can actually maintain both their structure here, and I will show you in a few slides their function. If we don't incorporate that, this is noted in the middle here, this peptide motif, we actually rapidly lose the differentiated, both the structure and differentiated function of the cells. That kind of makes sense, because what this is showing us is that the cells are actually able to interact with the hydrogel, and so they're actually both able to interact and sense what is going on around them. And as our controls, we use what is a standard system within the field, Matrigel, which actually has also a similar elastic modulus to the liver as well. But what we're going to show later on is, as opposed to Matrigel, we're not, then succumbing to some of the reproducibility issues that are often seen when using a natural hydrogel. So what we then looked was at using quantitative real-time PCR was to see whether or not these peptide hydrogels are actually able to maintain the differentiated function of the cells. And so we looked at a variety of different domains of hepatic function by quantitative real-time PCR. We looked at a variety of transcription factors where we found they are more robustly expressed and maintained as compared to traditional two-dimensional model systems. We see that there's a significant improvement in expression of transporters and nuclear receptors, as well as a variety of cytochrome P450 enzymes. And these are markers or domains that are traditionally been very hard to maintain their differentiated function in vitro. And basically, these domains that we're looking at here are expressed at levels similar to what we see actually in vivo for primary human hepatocytes. And actually, all the data I'm showing you here is for primary human hepatocytes. So now, it's one thing to just show that on the RNA level that we're actually able to maintain the differentiated function of these cells, but these are actually functional activity assays run on these gels. And so what we find is that for several different cytochrome P450 assays, we actually are able to maintain the differentiated function of these cells at levels similar to that of the most robust 2D system that has been engineered. And we show that here for BFC, noted on the left, which looks at a variety of different cytochrome P450s. But then when we drill down and look at very specific, mature adult cytochrome P450s, such as CYP1A2, CYP2A6, and CYP3A4, we find that they're actually maintaining the cytochrome P450, a very mature marker of differentiated hepatocyte function, and one that's very, as I mentioned, very hard to maintain at levels very similar to that of what's seen in our very robust 2D system, and at levels similar to a gold standard in Matrigel. It's not just that they express these cytochrome P450s, but there's actually a lot of machinery within the liver cells that enable the liver cells to respond to a variety of toxins or other substrates or compounds in vitro or in vivo. And what we show is that when we dose these different compounds in vitro, such as omeprazole or rifampin, here noted on the left, we see an expected and appropriate level of induction of cytochrome P451A activity in vitro. When we look in a different assay, looking at CYP2A6, we similarly see a significant upregulation in the presence of omeprazole and rifampin. And one of the hardest challenges is maintaining CYP3A4 inducibility in vitro, and we see we can do that as well, both with, as denoted here with omeprazole or rifampin here for CYP3A4 induction. And you can see that the levels that we induce are actually even superior to the best model gold standard systems, both in 2D or 3D here, denoted here on the right here for rifampin and CYP3A4. So basically, if we were then to summarize what we've shown here, is first of all, that we can actually generate a synthetic peptide hydrogel that can create a 3D microenvironment that has a similar elastics modulus, similar to that of the native liver. And moreover, we can functionalize this hydrogel to incorporate binding motifs which enable cell-specific binding and cell-gel interactions, which is quite important. And basically, using these synthetic peptide hydrogels, we're actually able to maintain hepatocyte survival, phenotype, and function over time, and that we assess this with very specific and very challenging methods showing that they can maintain cytochrome P450 activity. And so, by doing that, we can now show that we can definitely model hepatocyte toxicity and injury and drug metabolism in vitro in a system that can maintain their differentiated function over time, and that in the future, our plan is to move forward and to use this to model more complex disease phenotypes in vitro. So now, with that said, we can now move on, and I will turn this over for the Q&A or question and answer session.
Thank you so much. Okay, it is now time for the Q&A section. So the first question today is, how long can you maintain the hepatocyte in the peptide gels?
Well, I'll take that question, obviously, because it's directed to me. Basically, I'm showing data here where we're actually able to maintain the differentiated function and phenotype, and as well as survival of these cells. And this data is out for 2 weeks. That's about as far as we've pushed the system in this data, but we have preliminary data now that we can go as long as 3 weeks in the system. And that's a real advantage, I think, of the platform, because historically it's been quite challenging to maintain differentiated hepatocyte function over time, and particularly for lengths of time going off to 2 to 3 weeks.
Thank you so much. Okay, the next question we have is, why is media needed to obtain the gelation of the hydrogels? What are the active components of media in this process?
It's Mitch here. I think I can take this question. So it isn't a necessity to use media to cause the gelation. You could use a calcium chloride solution if you wanted to. I mean, effectively, what's causing the gelation is the divalent cation that's causing the gelation process, and these materials are commonly found in most types of media. So it isn't specifically media; it's the divalent cations that are needed in order to cause the gelation process.
Thank you so much. It's really interesting. Okay, the next question we have is, the change of the pH to interior of the cells does affect the fabrication of tissues, and if affected, how do you measure it? Hello. The next question we have is, the change of the pH to the interior of cells, does this affect the fabrication of tissues? And if affected, how do you measure it?
This is Matt. I'm not the right person to answer this question because I think it's probably one for the Biogelx themselves. But I guess this is gels rather than cells. How does changing pH to the interior of the gels affect the fabrication of the tissues? I'm not sure how pH dependent the gelation process is, Mitch.
Yeah, so the pH or the gelation process, I think one of the advantages of the synthetic materials is that the gelation process is completely independent of pH and temperature. So I don't, in terms of the gelation and forming the gels to mimic tissues, then that's independent of pH. It's neutral.
Thank you. Okay, the next question is, I see that you use Matrigel as a control gel for these studies. How could hepatocyte in other gel systems such as collagen compare to the peptide gels?
So although so this is Rob Schwartz. So although we didn't include this data here because we tried to create, include one gold standard in the data that we presented using Matrigel, we have compared them to collagen gels as well as PEG hydrogels. And so basically one of the challenges with these different gel systems is that the elastic modulus actually significantly exceeds the elastic modulus that's found in the liver. And hepatocytes are actually quite sensitive to the elastic modulus that's present within the hydrogel, and they actually will have significantly altered both phenotype and function in both collagen and other, higher elastic modulus containing gels, and so, or engineered gels. And so basically that was represented in the data when we looked at collagen with upregulation of a variety of dedifferentiation markers and other markers suggesting that the cells are under significant stress.
That's great, thank you. The next question we have is, why did you think these gels would be good for looking for bioactive metabolites?
Well, when we wanted to look at for the bioactive metabolites, when we wanted to try and see if we could find metabolites that would change the way the stem cells behave, we wanted a material system that was clean, that itself wasn't biological, which would contain lots of different metabolites and proteins, but was one that in each situation, so each of the situations that would drive differentiation, so in this case we use stiffness, was otherwise as identical as possible. So we didn't add artifact into the hits that we got from the mass spectrometry. So if you use like matrigel, you're going to have hits just from the matrigel itself, never mind what the cells are doing. So it was really important in the situation to use a synthetic gel.
Thank you. Okay, the next question we have is, can the cells grow in functionalized hydrogel by stained in situ by immunofluorescence?
So I guess either Rob or myself could handle this one. I'll go first and we'll see if Rob agrees with me or not. But yes, you can stain the cells in situ. You have to be somewhat careful about what filter you choose because the Fmoc group can give a fluorescence. But yeah, we've stained cells in situ lots of times. I don't know if you have also done so, Rob.
Yes, so I actually would echo what you just said. I mean, as long as you stay away just from kind of the autofluorescent spectrum of the gel itself, we were actually able to stain the cells in situ, and we've done that for a variety of different proteins without any difficulty.
That's great, thank you. I think we have time for one more question today, and the last question for today is, why did you choose to use parasites rather than MSCSs?
Okay, well, thanks for that question. We chose to use parasites because we were interested really in this possibility that cells within the gels could support hematopoiesis. And parasites are an earlier phenotype, if you like, than MSCs, and also more pure cell population. So we wanted to look at this expression of neural proteins that Adam Engler and Dennis Disher had seen and see if it could be used to control the bone marrow niche phenotype of the cells, see if they could help with hematopoiesis. And so that's why we chose to use parasites rather than mesenchymal stem cells, just to see if we could create a more bone-like niche environment. And we think that the gels have helped us to do that.
That's great. Thank you so much. So that's all we have time for today for the Q&A, but any questions that we haven't had time for, you can always email if you do decide to. So that just leaves me to thank Robert, Matthew and Mitch for what was a great presentation, and to Biogelx Limited for sponsoring this session. To the attendees, you will receive an email shortly telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is www.business-review-webinars.com. We look forward to sharing further presentations with you, so please do keep an eye out on our website and follow us on Twitter at BRWebinars for daily updates, and join our LinkedIn group, Business Review Webinars. Thank you once again, and I hope you all have a lovely day.